So our project is about the anthropogenic diet of four-doored coyotes causing an altered microbiome. My name is Rachel.
My name is Madeline.
My name's Angeles.
My name is Tamara Gallery.
And my name is Negin Paramonzadeh.
The gut microbiome is important to an animal's health because it can affect the animal's metabolism and immune system. According to one of our sources, Frontiers in Physiology, Martin and Associates stated, a clear relationship has now been established between gut bacteria and host metabolism, in which microbial-mediated gut hormone release plays an important role. An article from the Harvard T.H. Chan School of Public Health stated that the type of food an animal consumes has an effect on the type of bacteria living in their gut. For example, some foods may increase or decrease the pH in the gut, which determines what bacteria would be able to survive or not. In an article from the Microbiome Science and Medicine, Amato's study stated that the gut microbiome has a lot to do with an animal's overall health, including their nutrition, immune system, and behavioral patterns. Based on previous research done by Sudgen and Associates, rural coyotes have a much healthier diet than urban coyotes due to their bacteria content in their gut. These bacteria can promote protein-rich diets and improve body conditions. Sudgen and Associates also stated that some of the bacteria found in rural coyotes are Fusobacterium, Sotorella, Anaerobiospirillum, and Allobaculum.
Our project focuses on analyzing bacteria isolated from urbanized coyote scat to determine if their diet is beneficial to their health. We will compare the bacteria that was isolated from our coyote scat samples to previous research from rural coyotes. We believe that the bacteria isolated from our coyote scat samples are going to show that urbanized coyotes are not as healthy as rural coyotes because of their anthropogenic diets.
The first step of our material and methods were isolation of bacteria. The strain was isolated from coyote scat found on a trail near Fort Ord in Salinas, California on Wednesday, January 22nd, 2020. There were 4 scat samples created that contained either fur, bone, twigs, or moss. Each sample was contained in a separate falcon tube with a nutrient broth added to each of them and were shaken until everything was evenly distributed. The falcon tube were incubated at 37 degrees Celsius for 24 hours. To isolate the colonies, the broth were stripped onto LB plates. From those plates, isolated colonies were then stripped multiple times to obtain axonic cultures. The plates were put through several different chemical and physiological tests. Next test we performed was gram stain. The gram stain was done to determine whether the bacteria was gram positive or gram negative. If the bacteria comes out pink after the staining is done, then the bacteria has a thin cell wall, meaning that it is a gram negative. If the bacteria comes out purple color, then the bacteria is gram positive because it has a thick peptidoglycan layer that the purple stain can adhere to. Next test was PCR. Following the incubation of our culture plates, we were able to incubate a colony of our bacteria using aseptic techniques. We used a DNA purification kit and PureTech ready-to-go TM-PCR-B's reaction tubes to prepare our samples for amplification. Using polymerase chain reaction, we were able to amplify our bacteria using 16S rDNA with 27 forward primer and 1492 reverse primer. After doing so, we send our samples to the molecular cloning lab in San Francisco. Next test was aerobic chamber. During this test, the sample was placed into an anaerobic chamber for non-exposure to atmospheric oxygen by using hydrogen and nitrogen in order to reduce oxygen level for the purpose of analyzing our sample without interference of oxygen. Next test was nitrate, which was done to determine if the bacteria is capable of denitrification. This is done by testing to see if there are any denitrifying enzyme present. If there's no color change after 10 minutes, then a pinch of zinc powder is added. There was a color If there was a color change to red after reagent A and B were added, then there was a reduction to nitrate. If the color changed to red after adding the zinc powder, then there was no nitrate reduction. If there were bubbles present in the Durham tube, then there was reduction to nitrogen gas. Next test was oxidase. We also wanted to determine if the bacteria had cytochrome C oxidase enzyme, so the oxidase test was also performed. If the strip turns purple, the bacteria has the cytochrome C oxidase enzyme, which means that it's capable of aerobic respiration by having oxygen be the final electron acceptor in the electron transport chain. Next test was indole. Enzymes were enzymes were analyzed to identify the bacterium's ability to produce different reductase. An indole test was used to identify the sample's ability to produce indole from the amino acid tryptophan to also use as a carbon source. If it cannot produce indole, it doesn't have tryptophan. We add about 5 drops of Kovacs reagent to observe a red or pink circle on top of the tube, indicating that it is positive for indole. Next and last test was amylase, which was done to determine if the bacteria is able to use starch made from glucose as a carbon source. If there's a purple color around the strix, then the result is negative. If there's a hollow or clearing around the strix, then the result is positive.
These are the PCR results that we got after blasting the sequences. We got Enterobacter, Shigella, Acinetobacter, Bacillus, and Escherichia. The two main frequent results we got were Acinetobacter being 35.7% and Escherichia being 38.1%. Alrighty. These are the biological tests. We did nitrate and the metabolic tests. So we use nitrate to determine if the bacteria is capable of reducing nitrate. So for the first step in the nitrate test, we added the reagent A and B to bacterial samples, and we have two possible test results. Red positive, which means that bacteria is fully capable of reducing nitrate to nitrite, and no color negative, which just means that there was no nitrite present. So then we added zinc to the same sample to see if there was any further reduction. And red negative means that there was no nitrate present, while no color positive just means that the color changed from red to clear, and if there was bubbles present, it just meant that nitrate was reduced to nitrogen gas. So for our results, we see that Bacillus, Escherichia, Shigella, and Enterobacter all are capable of reducing nitrate to nitrite, but Acinetobacter is the only one that was not capable of reducing nitrate to nitrite or nitrate to nitrogen gas. For the metabolic tests, we analyzed oxidase, anaerobic chamber, indole, and amylase. Each color represents a different bacteria: Bacillus, Escherichia, Acinetobacter, Shigella, and Enterobacter. And if they tested positive, they would land in 1. and if they tested negative, they would land in -1. For anaerobic chamber is a little bit different, just because when using this test or when performing this test, you either determine if it survived or it didn't. So if it lands on 1, it just means that it survived. But if it were to land on -1, that just means that it did not survive. For the oxidase test, Bacillus was the only bacteria that tested positive, meaning that it contained cytochrome C oxidase, an enzyme making it capable of aerobic respiration by having oxygen as its final electron acceptor in the electron transport chain. In the anaerobic chamber, all the bacteria we analyzed survived, and they are capable of living in the absence of oxygen. And for the indole test, the only bacteria that tested negative, just meaning that it's not capable of making indole, are Bacillus and Acinetobacter. The rest can produce indole from amino acid tryptophan to use as a carbon source. All the bacteria except for Bacillus and Shigella had negative results for amylase test. This indicates that Bacillus and Shigella are the only bacteria capable of using starch from glucose as a carbon source.
So Fort Ord Monument is a large piece of land that is about 28,000 acres and extends from Monterey to Salinas. Its many neighboring cities are largely populated by humans, and many of the surrounding land is used for agriculture. The land is prone to wildfires, such as the river fire that happened last year in 2020, and it may cause many animals, including coyotes, to find food and shelter near urban areas. The coyotes of Fort Ord have been urbanized to frolic around homes, campuses, and anywhere humans may reside to obtain the necessary foods they can access. Due to the pandemic, there has been an increase in hikers around Fort Ord. These hikers have been leaving behind their trash and waste, which is adding to the anthropogenic foods available to these coyotes.
So when comparing the rural coyotes to urban coyotes, the common dietary component of rural coyotes contain higher protein levels from consuming their prey and other natural foods. Some examples of their natural foods include red berries, birds, deer, and brush rabbits. Urban coyotes located here in Fort Ord have a more diverse diet compared to rural coyotes, since they're able to consume more anthropogenic foods that are rich in carbohydrates. Some examples of the anthropogenic foods include sources from food from garbage cans, fruit trees, compost piles, and bird feeders. Coyotes are carnivores, making them hindgut fermenters, meaning that they have a single chamber stomach. And in this chamber, cellulose is broken down with the help of symbiotic bacteria. Most of the microbial fermentation occurs in the small intestine, followed by the large intestine and the cecum. According to one of our sources, Microbial Ecology, studies show that the dietary components such as proteins, lipids, starches, and simple sugars are absorbed in the small intestine, where pH is lower, transmit time is shorter, cell turnover rate is higher, and conditions are more aerobic. Since the coyote microbiome favors anaerobic conditions, there is a wider variety of taxa that can be absorbed in the coyote gut. The taxa present in the rural coyotes include Fusobacterium, Shutterella, Anaerobiospirillum, and Allobaculum. And the taxa present in the urban coyote gut include Escherichia, Shigella, Acinetobacter, and Bacillus.
So these are the bacteria found in urban coyotes. These are the bacteria that we decided to focus on based on their frequent appearance in our results. which are Escherichia, which is basically E. coli, Acinetobacter, Shigella, and Bacillus. So, in the gut, there are parts that may have less oxygen, and specific bacteria may thrive more than others in these locations, for example, in the small intestines. So, to determine if the bacteria is capable of surviving in these areas of the gut, we analyze the oxidase and anaerobic chamber results. As we saw from our previous data, all the bacteria, E. coli, Shigella, Acinetobacter, and Bacillus, um, are all capable of living in the absence of oxygen. But one particular bacteria, Bacillus, contains the enzyme cytochrome C oxidase, which allows them to survive and use energy. This just leads us to believe that the bacteria are facultated anaerobes, which just means that they are capable of surviving with and without oxygen. And Bacillus, being the only one with cytochrome C, can create ATP, which allows them to survive differently than the rest. And then to analyze the potency of the bacteria in the gut of the coyote, we observed the results from the indole and amylase test due to their biological functions in the intestines. Um, from our indole results, we noticed that E. coli and Shigella both produce indole. And from our research, we found that in the gut, indole is a signaling molecule that promotes an epithelial barrier to prevent the invasion of pathogenic bacteria. So while researching the bacteria, we already kind of assumed that E. coli was a prominent bacteria in the gut, just because it helps balance the amount of bacteria in the gut, but we weren't sure about Shigella. Shigella can be pathogenic, but if it were to have if the gut were to have a normal hefty amount of this bacteria, it would be considered healthy since it does produce indole to induce that barrier. We then analyzed the amylase test results, and we found that Shigella and Bacillus use amylase to break down starch and turn it into simple sugars. These simple sugars can be used for energy for the body to use in this case, for the coyote stomach. Um, the stomach absorbs the energy and it uses it to move the stomach muscles, which can promote good digestion. We were also not sure about Bacillus being a good bacteria, but again, it's similar to Shigella, where a certain amount of could be healthy when there is a balanced amount of bacteria in the gut. Finally, we analyzed the nitrate test results, and as seen before, Escherichia, Shigella, and Bacillus can reduce nitrate to nitrite. We thought this was important because sometimes the amount of nitrate in the stomach may cause stomach acid to decrease in pH or become more acidic, and this may cause harm to the coyote stomach. But because it can have this bacteria that reduces nitrate, the amount of nitrate may reduce and prevent a decrease in pH of the stomach acid. Overall, we concluded that Escherichia, Shigella, and Bacillus can be healthy in the gut in normal amounts, but if the amount of bacteria in the gut becomes unbalanced, it may provoke pathogenic bacteria to invade and prevent digestion and absorption of nutrients, such as Shigella and Bacillus, which can become pathogenic. The anthropogenic food that coyotes eat may cause this imbalance in the gut, and that's why urbanized coyotes don't have proper nutrients in their system. It's being blocked by pathogenic bacteria. We also noticed that Acinetobacter does not provide any support or health benefits to the gut of the coyote. It did not test positive for the indole nor amylase test, but because it did survive in the anaerobic chamber, it can live in the gut or in particular places where there's a lack of oxygen. Um, from our research that we gathered, Acinetobacter also is very resistant to antibiotics, and it can live in hospitals. From an article that we found, the scientists talked about Acinetobacter being present in veterinary hospitals. They did further studies and isolated samples from their furry patients, which are cats and dogs, and found that many were infected with this bacteria. It was found in the urinary tract and the respiratory tract, and it could also be found in other locations. They were developing symptoms, and it's really hard to treat because it's very resistant to antibiotics. This led us to believe that if coyotes were traveling to urban areas where cats and dogs are located, they sometimes also tend to eat them. Um, Eat dogs or cats, they may be contracting that bacteria from these pets and locations. But it can also be found in the soil and food, which is where they mostly get their nutrients from, possibly um, in the garbage. Also, garbage found in hiking trails, which has increased due to the pandemic.
To recap, when comparing the bacteria that was isolated from urbanized coyotes, Escherichia and Bacillus were able to survive without oxygen and have cytochrome C oxidase. This allows for the bacteria to produce ATP more efficiently under aerobic conditions, making them facultative anaerobes. Shigella can also survive without the presence of oxygen and is capable of converting complex carbohydrates to simple sugars that can be used to produce energy for the coyote to digest its food. We discovered that Acinetobacter is the most harmful to the coyote's health since it does not have indole or cytochrome C oxidase, which are components necessary for its survival.
So, in conclusion, Escherichia and Bacillus have components that promote respiration and can be beneficial to the coyote's overall health. Shigella is also beneficial to the coyote's health since it's able to produce energy for the coyotes to digest its food, which can be very helpful for the coyotes with diverse diets. Finally, Acinetobacter is the most harmful to the coyote's health since this bacteria does not have any of the components necessary for its survival. With that being said, if there's an imbalance between the different bacteria that are found in the coyote's gut, some may be more harmful than others, in this case, Acinetobacter, and can lead to pathogenic diseases. Urbanized coyotes have a more diverse diet compared to rural coyotes, which leads them to ingesting more bacteria that can be harmful to their health. Although this is the case, having a more diverse diet allows for the coyotes to intake more anthropogenic foods since the foods in this diet is more constantly available. Coyotes are known to be opportunistic species since they're able to colonize and adapt to new environments by exploiting diverse food sources. This enables them to thrive under unpredicted circumstances when natural foods are less available.
Okay. In terms of our future work, these are the questions we would like to further investigate when doing this type of research. How can we contribute to the coyote's survival? How can we strive for a better environment and promote good health among the coyote population? What aspects of the anthropogenic food are causing coyotes to develop some forms of gut bacterium? So during our research, we realized that there was not much information or research about rural coyotes and their microbiomes. It would be interesting to read about more research being done to study the diets of coyotes and how it relates to the bacteria found in their microbiome.